Hey guys, welcome to our third topic in Chapter 5, How Viruses Replicate. This is a really important topic because this is what's going to help set up the stage for how viruses invade our cells and how they replicate within our bodies. So why is this topic important? And viral replication is the key to viral infection. So it's really important that we understand how the viruses get into our cells, what they do to our cells, and then how they leave our cells. We're also going to have a few slides at the end of this lecture about some lasting effects that they can have on our cells. So pay attention to that part at the end as well. So here are our objectives for this section. These four objectives seem pretty short, but they hide a lot of information within them. So make sure that you understand all the parts that these objectives are asking for. And as always, if you have any questions, feel free to holler at me, email me, ask in class, and we'll go over any of these objectives. All right, so let's get started with modes of viral replication. So viruses are all about viral replication, and they're little infectious agents. They're really, really small. So they have a unique method of invasion replication, and every virus is slightly different, but we're going to talk about some of the big pathways and the big ways that these things happen within the cell. And so it's really important to not get hung up on specifics. We're just looking at the general overview here, especially envelope versus naked viruses. There's some differences in that as well. So let's start with the cycles of animal viruses. So there's about 5 steps to the virus replication cycle. And just like I talked about in chapter 3 with that prokaryotic cell, this is the same thing. This picture sums up this part of the topic. So make sure you understand everything they have here. And I know there's a lot of information in each of these steps. So take your time, work through them, put them into your own words so that you can make sure you understand them. And remember, this is all variable virus to virus, so we're just looking for the general principles. So the general phases of viral replication in animal cells is absorption, penetration and uncoating, synthesis, assembly, and then release. And we're going to go through each of these on the following slides. So absorption. Absorption is how the virus starts to enter the cell. Remember those spikes we talked about in the last section? Those spikes will key into specific receptors on the cell and help them become absorbed within the cell. And these key locks and keys that these viruses in the cells have, they can vary depending on the cell. So for instance, this changes the types of cells that viruses can target. Like for instance, we know HIV targets white blood cells, and that's because of this lock and key mechanism. You see a couple other examples on the slide here with liver cells and intestinal cells, etc. And so that's why it's important that these things are really specific for each other. And this is also, to an extent, how viruses are species specific because of these specific receptors. So it's really important to recognize that host, range, and tropisms are the key to virus absorption. So penetration and uncoating, this is how viruses actually get into the cell. And there's 2 ways that viruses will enter the cell. The first is through endocytosis, and the second is through viral fusion. In endocytosis, that's the first picture you see on the slide here. The virus will attach or absorb, and it will be brought in within a vesicle within the cell. And it will then, once it's in the vesicle, this vesicle will break down and release the viral DNA or RNA into the cell for replication. And this is through an uncoating process. Now, when viral fusion occurs, that envelope actually fuses with the host cell membrane, becoming a part of the membrane. And then that will release the nucleocapsid into the cell, and then the capsid will be uncoated, releasing that genetic information into the cell. So make sure you understand how the 2 work. In one, the envelope becomes part of the cell membrane, and the other, it is absorbed into a little pocket that's brought into the cell. So synthesis. This is the big game, right? This is what we're all after. This is the replication and protein production. Remember, we have two main types of nucleic acids we talk about, DNA and RNA. And in this case, we're also going to break out retroviruses into their own category. So we have DNA viruses, RNA viruses, and retroviruses. And we're going to look at an example of double-stranded viruses. There are two phases of replication for double-stranded viruses, the early phase and the late phase. And we've kind of hit on these already in the earlier topics, but bear with me as we go through them again. So in the early phase, the viral DNA is going to enter into the nucleus, and this is where the genes are going to transcribe it and make that messenger RNA. Messenger RNA is then going to be sent out of the cell to the rRNAs and the tRNAs, and they're going to transcribe this information into the proteins. And then the proteins are going to start getting made. And so we have this whole process going in. And this is part of the this transfers into the late phase at this point in time. So then once we have new copies of the genomes, new copies of the capsids ready, they are released through budding into the cell. So that's really a brief overview. So remember, the DNA gets in, it's copied, some of it's made into RNA, so it can be sent in for protein manufacturing. Some of it's copied for future viral particles. It's all brought together in late phase, and then it's released out of the cell, either through budding or through other processes. So how did we get it assembled? There's a bunch of different assembly methods, and once again, it comes back to how are these viruses working out. And this is a really good picture for you because it's very visual on how this process works. And you can see how the positive RNA sequences work versus the negative. I just want you to have some generalized overviews on this. You don't need to get too specific, but be familiar with how these viruses work, especially the RNA versus DNA. And remember those sites of transcription and translation. Okay. So once the virus is assembled, it's time to release. Now there's a lot of different ways that viruses are released from the cell, but we're going to look at viral budding. So what you see here in the viral budding is we have the capsid and we have the RNA, and the capsid comes around, protects the RNA. It then attaches to the cell membrane where it then will bud off, and this creates a new virus envelope or new envelope virus from the cell. And remember, this process only works for envelope viruses. Okay, non-envelope viruses have a different method of escaping the cell. So be sure you're clear on that because budding has to bring the membrane with it. Okay, and so once it's released, it will go to other cells and start infecting other cells. So this is how viruses will spread within the body. So let's take a moment and review our steps. We have absorption. This is where the virus will find cells that have the right lock for their key. We have penetration and uncoating. This is how the cells are brought into how the viruses are brought into the cell, either through envelope fusion or through endocytosis. We then have synthesis, where the DNA and the proteins are made. We have assembly, where the DNA or DNA or RNA, the genetic information, and the proteins are brought together to assemble. And then we have release, which is where, in this case, this specific case, an envelope virus will receive its envelope and leave to go and infect other cells. So make sure you really have a good handle on those. So in these last four cells, we're going to talk about the damage that that these viruses can do to our cells. And the first of these is talking about host cell damage and persistent infection. One of the easiest ways to see host cell damage is through the cytopathic effect, or CPE. In this case, the virus induces damage that alters the appearance. And you can see that on the picture on this slide. You see all those rounded up cells? That is cytopathic effect versus the cells that are nice and long and striated, kind of on the right hand side of the picture. That's more normal cells. So that's what we're talking about when we talk about CPE. See how there's a gross physiological difference between the types of cells? That's what viruses can do. There's also persistent infections, and this is where the cell harbors a virus due to the virus not being lysed immediately. And so the cell will maintain these infections, and they can last from weeks to a lifetime. And this is kind of how chickenpox works with varicella zoster. And this is also sometimes known as the chronic late stage persistent infection. Another example is proviruses, where the viral DNA is incorporated into the DNA of the host. And so every time this cell is then replicated, this DNA stays with that cell. And that's going to be really important in this next topic when we talk about viruses and cancer on the next slide. So it's been in the news a lot lately where we talk about like HPV vaccines and HPV in general, which the human papillomavirus, which can cause cervical cancer, and also it can cause throat cancer. So if it gets into your throat, and what it does is it can alter the genetics of your cell. And so there's three different ways that viruses can cause cancer. We have some of the retroviruses, the oncogenes incorporate into the DNA and produce the proteins that lead to uncontrolled cell growth. And then we have other ones where the viral genes affect the expression of oncogenes within the cell for uncontrolled growth. So we have one where they make proteins that lead to uncontrolled growth. We have some where they change the way the proteins actually work within the cell. And then we also have viral genes, which directly regulate the way the cell works and how it actually just causes it to have uncontrolled growth. So all of these ways are ways in which tumors are produced within the cell. And about 1/5 of human cancers are caused by viruses, but we're still discovering a lot today. So who knows? There could be a bigger component of this, or we may have identified all the ones that are viral associated. So make sure you spend a little bit of time on this, but it's not the biggest thing in the world. Just make sure you understand how viruses can cause cancer cells within the human or within animal cells. So now let's talk about viruses that affect bacteria. Most of this time we've been talking all about how these viruses are impacting animal cells, specifically us. But some viruses actually infect bacteria, and these are called bacteriophages. And you can see here on the slide we call them T even, and that's because we have T2, T4, etc. Bacteriophages, and they just are slightly different, and so that's why as a class we call them T even. And this is where you can see that complex capsid that we talked about back in topic 2. They have a cosahedral capsid head, the central tube, the collar, the base plate, tail pins, and fibers, and they actually inject their nucleic acid into the cell. The actual virus protein does not enter the cell in the case of the bacteriophage. So let's look at how this life cycle works for bacteriophages. So in this process, we have the lysogenic cycle and the lytic cycle. And so what you can see here is in step 1, we have absorption. This is where the T bacteriophage binds to the bacterial cell. Penetration is when they inject their DNA or RNA into the bacterial cell. This then gets replicated within the bacterial cell, and this is all still part of the lytic cycle. And then new proteins and new copies of the viral genetic information are made, and then these are reassembled within the cell, and then the lysis of the cell occurs, and the bacteria will actually, or the bacteriophages will escape from this cell to go on and infect more bacteria. So it's really similar to what we've been talking about with the steps of the lytic cycle. Now, in the lysogenic cycle, the viral DNA is inserted into specific sites in the viral chromosome, and this viral DNA is duplicated along with the regular genome and can provide adaptive genes for the host. And this can actually create new diseases within the human body, or within the bacteria, such as antibiotic resistance even, or the production of toxins that can make us sick. So through a viral infection of the bacteria, it can change the bacteria so that it then impacts us as far as human health goes. So it's kind of crazy that way. Bacteriophages are a really big growing area of research right now, so you'll probably hear a lot more about them as you progress through your studies. But as far as we're concerned in this class, we're going to focus on animal viruses, but be aware of what the bacteriophages are and how they work. And I have a couple videos on the lytic cycle and the lysogenic cycle up for you on Blackboard, so you can check those out if you want to see some visual examples of that. And I also have some ones on the other cycles too. So I know these are really complicated and I feel like we went through them a little fast, so make sure you go back and you really review those steps. So remember, we have absorption, penetration and uncoating, synthesis, assembly, and then release. Those are the 5 steps that are really important to understand. And remember that to an extent that it plays a big role as to whether or not that viral DNA or RNA is incorporated into the host cell, as to whether or not it results in a persistent infection or even cancer cells. So be aware of how that works. That's really what I'm looking for, is some overall conceptual understanding of how those processes work. And as always, review your objectives, let me know if you have any questions, and shoot me an email or ask me in class.